Well, good morning, everybody. Really great to be here. What I thought I could provide over the next couple of minutes is really trying to connect what you've heard through the last few days on genomics and its connection to cancer, but maybe do that from a technologist's viewpoint. And I think it's maybe a viewpoint that can get at the question that all of us really want to see answered, which is, when will this genomic revolution in medicine actually take place? Now, there's a fair amount of cynicism, perhaps, in the general society. It's been 10 years since the first genome was sequenced, and when you read articles that are in the Business Week or in the newspapers, the question is, what do we have to show for it? Well, again, what I want to show you is that over the next couple of minutes, we've actually come quite far over the last decade, and we're actually quite close to answering this question to really impact patient outcomes. Now, as Mark said, I was here last year, and we shared with you a vignette of a notional patient in the future named Sarah that was predisposed to breast cancer. But she was a fortunate one that she was living a few years from now and met her oncologist who was trained in molecular methods. She was sequenced by machines that I told you would get smaller and smaller and smaller, more out to the doctor's offices. And because we could read her encyclopedia of life, if you will, we could find those mutations on her DNA and prescribe her the right therapy. And she would be a story of survival, and hopefully one of the 40,000 women today that aren't a story of survival with breast cancer. So that was really what we set up in terms of how will we make that genomic medicine revolution take place. Well, maybe first to talk about that revolution from a technology standpoint. So the machines have come a long way, but actually, even in the last decade, they still use cameras. So to explain to you actually how you read a genome, you take the DNA in a sample, you run it across a lens, and then you open the shutter millions of times where you take a picture of a chemical reaction. And then, like you probably have done on vacations, you piece together those photographs, and then you come up with an image of somebody's DNA. The problem with the camera is it's expensive, and you can hear by my explanation it's fairly slow. And more than that, you'll never move from the research bench and kind of one-off genomes or hundreds off genomes to the millions of genomes that have to get done out into the medical clinic. And in order for that to take place, it's got to happen in hours and minutes where you can read somebody's genome. Well, the news here today, and kind of the new stuff from last year, is that we have made enormous progress in the last 12 months. And actually, what I want to share with you this morning is something incredibly groundbreaking called ion torrent genomic sequencing. This is a pictograph, an image of actually now direct sequencing. No more cameras, actually just directly reading the DNA on a silicone chip. Actually, the hydrogen that comes off when you read your DNA base by base. And as you can see, it's using semiconductor technology, which allows you to leverage the trillions of dollars of investment that's gone into the electronic age now into genomic medicine, potentially. And if you think about what's taking place here, this chip actually that we're showing you, that I'll show you actually in a moment, uses 1995 technology. We actually found a foundry in Taiwan that still makes chips from the 1995 vintage. They're in your car, actually. But those chips have wells in them that are farther apart. We wanted to prove the principle first before we would go to the mass parallelism of modern-day chips. But just kind of think forward. If you move to the year 2000, you'll be able to put these wells closer together. And reading a genome, which today, in the modern, most advanced machines, takes about a week, you can take this thing down into about 2 hours. Or if you go to 2005 technology, it gets even more dense. So what I want to share with you now is something that is entirely brand new, hasn't really been shown yet. This is the personal genome machine. This is just being launched to general users. If you look at the size of it, it's not very big. It could go into a doctor's office. The screens are incredibly easy. Literally anybody in this room could use it. You go up, it's 6 buttons you press in terms of questions they ask you about the sample you put in. And you don't have to stand by it. It actually uses an iTouch that allows you to monitor the sequencing of the genome. And this will allow the sequencing of the genome to finally take place in the question of minutes and hours that takes it out into medicine once and for all. So the technology, what I would tell you, is not going to be the limiter to making that question get answered of when will the genomic medicine revolution finally take place. And I think that's why we're starting to see just incredible interest in general society of, who are we? The curiosity of our genome and what it gives us insights on and who we are. And you'll hear from the Osborns here shortly about their own journey of learning their genome. But perhaps even having more impact is what can it do in the clinic? What can it do if we have disease and solve it? And so what I want to share with you now is actually taking this technology and then how do you apply it? So I thought this would be a good image to share with you is actually how it's being used today. Now this is at the Baylor Genome Center in Texas. It's one of the largest genome centers in the world. You can see it has a bank of our machines in it. And this is 24/7. This is like a factory. They are doing thousands of samples. It's teamwork. And they're trying to figure out what gene does what. And that's a lot of work still ahead of us to make all of these associations. But genome centers like the Baylor one or the one in Boston, the Broad Institute, I mean, the scale at which they're doing this is profound in terms of research. And we are finding each and every day new gene associations, as I'm sure you've heard over the last couple of days. But that's research. The question really is, how do you pivot to medicine? Well, to pivot to medicine, you need pioneers. And you need physicians who want to be out in front, in the frontier, really practicing this stuff. And let me just share with you a few people, actually, that are doing it today. One of them you saw, actually, in the Stand Up for Cancer is one of our very close collaborators, Dan Von Hoff at TGen in Phoenix. We're doing a clinical trial with Dan to sequence many women who have triple-negative breast cancer. They've exhausted all other ways to potentially survive. Well, we're sequencing them, and we hope to have insights on what therapy will work for them. And our hope in the next year or so is to be able to show these results, and we hope that they will show an entirely new way of treating breast cancer. Dr. Lipsky in Texas found the genes that cause CMT disease, a disease he personally had, and is now developing the medicine to actually treat that. And then our own Eric Topol, as Mark mentioned, right here in San Diego, one of the world's premier cardiologists and now genomic medicine doctors. The interesting thing with Eric is that a few years ago he started this symposium called The Future of Genomic Medicine at Scripps here in town. And he got some comers from around the country that people were interested in taking their medical understanding to the next level in terms of using genomics. A couple years later now, you can barely fit them all in the auditorium. So it shows you that sometimes humans have a hard time seeing momentum. There is incredible momentum now happening in the medical profession to learn genomics and apply it to patients. So through the eyes of these pioneers, the question then is, what will the clinical interaction look like in the future using this technology to solve an individual's disease? Well, this is a vignette really from their eyes of how it would take place. This would be an oncologist who has now been trained in this college we've just spoken about, certified in the use of genomic methods to treat cancer. A particular patient has been genomically sequenced, both their native DNA and then the DNA of their tumor. The physician is able to call up all of that information quite readily. They're able to see the data, sitting down with the patient and going through what the genome has really given insight for. And then they're able to hopefully be able to compare what they've just seen to what available medicines are being either developed or on the market that could potentially treat, in this example, this gentleman's prostate cancer. That's how it will look. And I will tell you that's only actually about 2 years off or so if we do it all right with these pioneers. Now, in terms of the treatment itself, this is really more of how I think medicine will have to be practiced in the future. Again, I'm not a scientist and I'm not a doctor, but we spend a lot of time with the people doing it. As you've probably heard over the last few days, cancer has been historically described by geography, where it is on the body. And that notion is quickly getting thrown out the window. All cancers basically will be in the future described by their pathway. And we learned that actually with Dan Von Hoff recently when we were treating a patient who had brain cancer And it turned out the most effective drug on that genetic mutation was a testicular drug. And it had a really big impact on that patient. And it showed you that actually the cancers are sharing similar pathways depending on where they are, or not depending on where they are actually. But a cancer is differentiated by the pathway itself. And so how it will work is that we'll move from 1 in 5 drugs in cancer being effective today, when time is of the essence, 1 in 5 doesn't cut it, to where hopefully it's a much higher percentage of success. And so we'll be able to really sequence a patient. We'll be able to then compare it to the drugs available or the ones in development, put that patient into a clinical trial if necessary, and hopefully be able to design a custom cocktail. It could be several different drugs that work on the several mutations we find when we sequence their genome. So the question really then I will leave you is, what can you do? You know, when you read the newspapers today, and perhaps I could have you have a little higher sensitivity to when you hear some drug has been disapproved by the FDA, you know, just realize that our society has become very risk-averse with the approval of new drugs. And certainly in some cases that's warranted. But always remember, those drugs went to trial because they did have an impact on some patients, a very positive impact. And so somebody has been left behind without a new effective treatment for them. In the case of genomic medicine, the FDA is going to have to radically change its science, because how we're talking about treating patients in the future really is not how they're equipped to set up approvals. And I'll give you an example. Each and every time you develop a new diagnostic for a gene, it has to go through an exhaustive clinical trial. We're in the diagnostics business. We go through it all the time. Now, when you're able to sequence the entire genome, both the genes that express themselves and the non-coding regions, I mean, it's just completely impractical that you would want to have to do a clinical trial on each and every gene. We'll never get this revolution to happen in the United States under that regulatory regime. Even more complicated is that we think cancers will be treated like HIV in the future. There'll be cocktail drugs. There'll be drugs that are mixed together to treat that mutation and this mutation. And they may come from different companies. And I will tell you, the FDA is very unequipped to do these more sophisticated cocktail therapies for cancer. And so my worry and my hope at the same time is that we can have a more enlightened conversation as a society with science to change the FDA and really make this genomic revolution happen, which I think will happen in the next couple of years. Thanks very much for listening. Thanks, Mark.